Okay, on page E3, we were learning about sacs, membranous sacs that store chemicals. If they're large, they're called vacuoles. If they're small, they're called vesicles. And we had talked about secretion vesicles playing a role in secretion or exocytosis of chemicals from cells. We had also spoken of food vacuoles that are formed after a cell phagocytoses or swallows up, engulfs, or also known as endocytosis, something, so it forms a food vacuole. And then we had spoken of lysosomes. And we said that lysosomes have this strange nickname, suicide bags, which we're going to get to in just a moment. We said that the lysosomes are formed from the flat pancake-like sacs of the Golgi complex. They contain digestive enzymes, and they fuse with food vacuoles. Now, we actually showed you a picture of this on page E11, on E11. And again, there are resources available showing you all this junk. If on my website, for example, you can click on let's see, on phagocytosis. I don't even know which video this is. And we can click it, and look at that. Ooh. And here's the lysosome. Everybody see the lysosome uniting with the food vacuole, digesting the food. Whatever can be used is absorbed into the cell, and whatever can't be is expelled out. So that's exactly what we were talking about. There's other videos. There's a lot of these things. And I put them there for the purpose of making this.
The substances taken in by single-celled organisms are often particles or large polar molecules that cannot cross the hydrophobic plasma membrane. Many single-celled eukaryotes employ endocytosis to ingest such food particles. In this process, the plasma membrane extends outward and surrounds the food particle.
All right. It's okay, I'm stopping anyhow. So anyhow, there are plenty of resources available, and the point is for you to use them, or your book, and all the other resources. All right. So we had talked about this picture last time. This is covered, I believe, in Chapter 4 of your book, or Chapter 3. And I wanted to raise this question. Since these lysosomes contain these digestive enzymes that are designed to digest what's in a food vacuole, we might ask, what do you think might happen if a lysosome containing these powerful digestive enzymes ruptured, releasing these enzymes into the cell? What do you think would happen to the cell? The enzymes would digest the entire cell. In other words, it would be, in a sense, committing suicide. So, in fact, if we look right just to the left-hand side, let's enlarge this. So it shows what's labeled a living cell, and these are the lysosomes. And if these lysosomes rupture, so they, all these hydrolytic or digestive enzymes are released into the interior of the cell, and the cell self-destructs. And here it shows that happening. It's labeled a dead cell. And this phenomenon of a cell basically committing suicide, the name that the books use is apoptosis. So that is the term that's used in your textbook in Chapter 3. And it's also the term used in many of the videos that I've linked. We're going to call it by the name autolysis, which is an easier word to spell and say and explain what it means. Autolysis, made up of two roots, auto and lysis. Auto means self. Lysis means, of course, to break apart. So literally, putting it together, it means to break apart itself. And so that's what it's called when the lysosomes rupture and the cell self-destructs. Now, I want to give you a few examples of when lysosomes actually rupture, causing the cell to break apart. So let's return back to page E3. And back on page E3, under lysosomes, on page E3, so we wrote that the lysosomes can act as suicide bags in programmed cell death or autolysis. And I gave you 3 examples. I'm actually going to give you 4. Okay, the first example I wrote is programmed cell death in the embryonic formation of fingers and toes. And our first thought is, what does that mean? So I want to show you a picture. This is a picture of a human embryo that's 5 weeks old. You don't have this picture, but it's okay. All right, so in this picture of a human, a baby, 35 days old, it's the end of the 5th week, look at its hands and feet. The hands and feet start out like paddles. There are no fingers or toes. It's all a solid mass of cells. So, and in fact, if we look down here, you're starting to see fingers and toes form. So how do they form? Literally, there are vertical columns of cells that are genetically programmed to commit suicide. So when a vertical column of cells, the lysosomes rupture, releasing those enzymes, killing the cells, and that creates a separation. And another vertical column of cells commits suicide, creating another separation. And that's how we go from a paddle-like hand or foot to fingers and toes. So this is known as autolysis, right, of breaking apart itself, or programmed cell death. And of course, the program for all this happening, when we say a genetic program, we mean the DNA instructions that are in the cell. So this is actually how it happens. Now, sometimes this genetic program is defective. If the genetic program is defective, the person might end up with, it may not form fingers or toes at all, and might be born with a paddle-like hands and feet, or maybe they'd have only 3 fingers instead of the normal 5 on each hand. In fact, it could even be defective where the person ends up with 6 fingers on each hand and foot instead of the normal 5 or even 7. So whether somebody has fewer fingers or extra fingers, all has to do with whether this is occurring properly. Now, one of the interesting things that when a baby is born, for some reason, one of the very first things that almost every mother does when holding that baby for the first time is they pull down the blanket around the baby and they count the fingers and toes. All right? If you don't believe me, ask your mother. Just ask. Say that the teacher said at school that it's very common for mothers to count the fingers when their baby is first born. Did you do that? And if anybody has children, is that true?
Yes, very true.
Well, the first thing you just want to see, are they complete? Are they all there? All right? So it's one of the first, you hold the baby, you look at their face, and then you start to pull down the blanket to see if they've got all their fingers and toes.
Yeah, and then you tell your family members, yeah, all.
Then they're all complete. So that's how that works. Now, sometimes whether somebody's born with the wrong number is because of a genetic defect, a defect in the DNA, which they might have inherited, or it could be that there's a defect in this happening because the mother, my battery just went up, because the mother during pregnancy might have been exposed to a harmful chemical. And so women who are pregnant have to be more careful about what they're exposed to because there are various chemicals that can interfere with this normal development. After all, these embryos are really small and very sensitive to chemicals. So there are examples of this. Again, I don't want to take up too much time, but...
That's a quick question.
Yeah, go ahead.
What about when you're born with that extra little digit and is that process of that DNA?
Again, the 2 major reasons why something is wrong is either a defect in the DNA or exposure to something in the environment. It's actually, I'll just go to my anatomy page. I know I have this. Let's see. you can...
A historic compensation deal has been reached for victims of thalidomide. British company Diageo has agreed to pay $50 million to the surviving victims in Australia and New Zealand. An 86-year-old World War II veteran helped negotiate the payout. The drug caused horrific birth defects in the 50s and 60s, and many survivors are struggling to pay mounting medical bills.
Barry de Geese was a thalidomide baby, born with no arms and deformed legs. Today,
All right, I'm just going to explain what's going on here.
Remaining thalidomide victims in Australia and New Zealand who will benefit from a new multi-million-dollar payout.
Interestingly, in the 1950s and 1960s, a number of women were prescribed a medication to reduce morning sickness or the nausea that commonly is most women will experience during the first trimester, first 3 months of pregnancy. Some women experience nausea the entire pregnancy. This medication was actually made available in most countries of the world, including Britain and Australia and Canada and so on. And they never tested the drug. They tested the drug on women. They found it reduced nausea, but they never tested to see what effect it might have on the babies. There were literally tens of thousands of babies born with deformed arms and legs because it interfered with this normal development. Interestingly, this drug was never approved in the United States. The United States Food and Drug Administration, or FDA, is very strict in its approval of drugs. So do you think this ever happens today where there are medications not available and approved in the United States, but available, let's say, in Canada or especially in Mexico, and people go to other countries or buy these drugs on the internet all the time? There may be a good reason why these drugs haven't been approved in the United States. The U.S. is very strict because, and basically the only American women who gave birth to deformed thalidomide babies like this are women who went to Canada to get that medication, which was available at the time in Canada. So there were, other than that, it was never approved in the United States. So various medications even can interfere with development. So basically, when women are pregnant, doctors don't want to prescribe anything to them. About all they can say you can take a Tylenol, and if you get an infection, they'll give you penicillin. But other than that, they won't really give you any medications during pregnancy because they're just always so worried that anything could interfere with the normal development of the baby. Okay, so back on page E3, we gave you the example of the role of these autolysis of the lysosomes in the embryonic formation of fingers and toes. A second example that I've listed is atrophy of the uterus during menopause. So let me explain that. First of all, what is menopause? Menopause is something that occurs at about the age of 50 in women. It's when a woman's ovaries stop working. Now, I made a little arrow, and I just wrote the following at the bottom of the page. All right, so I drew an arrow here, and you could write what I wrote here. All right, so what did I write? At menopause, you'd say, yeah, what's menopause? Menopause, it occurs at around 50 years of age in women. It's when her ovaries stop working. Now, unlike a guy whose testes function his entire life, he produces testosterone, the male sex hormone, and produces sperm his entire life. A guy could be 97 years old and still father a child. Women's ovaries stop working at about the age of 50, sometimes a couple years earlier, sometimes a couple years later. Now, when a woman's ovaries stop working, well, we mean they stop working. They stop secreting estrogen, the female sex hormone, and they stop ovulating. They stop releasing eggs. So this is known as the reproductive biological clock in women. It doesn't exist in men. This actually makes women more serious about their life than men. I've talked about that before. Remember, a 20-year-old woman is getting serious about her life. A 20-year-old boy has clueless as far as what life is about. All right? So what happens, therefore, is the drop in the estrogen hormone level. Now, this drop in the estrogen hormone level, because the ovaries are no longer producing estrogen, causes a number of changes in a woman's body. And it causes changes in her skin. It causes changes in her bones. Her bones become more likely to develop osteoporosis, weakness of the bones. And it causes other changes. Now, one of the other changes that it causes, women don't really care that much about, but that's why I'm mentioning it. The drop in the estrogen hormone level triggers or causes autolysis in the cells of a woman's uterus. You'd say, what was autolysis again? It literally means to break apart itself. The lysosomes rupture and the uterine cells self-destruct. So the whole uterus, as a result, atrophies. The word atrophy means to shrink. So the uterus shrinks because a woman is not going to carry, she's not going to get pregnant now, she's not going to carry a child because her ovaries don't work anymore. All right? So the uterus shrinks. Now, most women really don't care that much that their uterus is shrinking. In fact, some women are almost relieved that they no longer have to deal with menstrual periods, which we'll get into later in the course. But the word atrophy means shrinking. You've heard that word atrophy. Some of you might say, I never heard of it. If you don't use your muscles, what happens to your muscles if you don't use them? They shrink. They atrophy. All right? So in this case, we're not talking about somebody's arm muscles atrophying or shrinking because of lack of exercise. We're talking about a woman's uterus shrinking. Interestingly, what they used to do up until about 10 years ago or so, 15 years ago at the most, is when women would approach 50 years of age, the gynecologists, which are doctors for women's reproductive issues, would prescribe estrogen. This was known as hormone replacement therapy. And they would prescribe estrogen, and that would prevent these changes from occurring. In other words, it would prevent the changes in skin and the changes in developing osteoporosis of the bones, and also it would prevent the uterus from shrinking, not that anybody cared. Now, taking estrogen doesn't make the ovaries work, so women still couldn't get pregnant after the age of 50. It just prevented some of the physical changes that occurred. However, about 10, 15 years ago, a large clinical study was done that showed that in a small percent of women taking estrogen after menopause, a small percent of women, probably less than 2% of all the women taking it, had an increased risk of dying from strokes and other cardiovascular problems by taking estrogen postmenopausally. For that reason, most gynecologists either stopped prescribing estrogen in postmenopausal women entirely or will only prescribe very, very low doses. So today, women can take other medications to prevent osteoporosis or weakening of their bones. You've heard the commercials done by Sally Field for Boniva and Fosamax and so on, but they no longer really prescribe estrogen. And that was because of some side effects that are found in some women. All right, but anyhow, the point that we're just talking about a lot of different things, but what's uniting all these things I'm mentioning is the role of lysosomes acting as suicide bags to cause cells to self-destruct. I want to give you a third example. And the third example I wrote up here is atrophy, which we now know means shrinking. Atrophy of a tadpole's tail during the metamorphosis into a frog. And our first thought is, what the hell does that mean? Let's turn back to page C9. Way back on page C9. And this was page C9. Now, way back on section C, page C9, so the background on this is that we were talking about, I believe on page C8, we were learning about radioactive isotopes. Is that right? On C8. And we had talked about radioactive uranium on page C8. And we had talked about radioactive carbon-14. And the third example we gave you is radioactive iodine. And we learned about the thyroid gland. And we learned that our thyroid gland needs iodine in order to manufacture a hormone called thyroxine. And we learned on C8 that thyroxine is a hormone that increases our metabolic rate. It speeds up the rate of biochemical reactions in a living thing, in us or in any other living thing that produces thyroxine. Now on page C9 there was this picture. Nobody ever asked me anything about it. And it had pictures of frogs mating and reproducing. And we might wonder, what the hell is that all about? So I'm going to explain it right now. The subject of metamorphosis is interesting. Clearly, it's an interesting subject for biologists. Because we know, And let's just start here at the bottom. So in the very bottom picture, it shows a male frog typically will get on top of the back of a female frog and ride her in the water. They do this in the water. Some of you, anybody ever seen like insects mating while flying? Okay, you'll see that with like dragonflies and so on. But anyhow, so they're in the water, kicking around, and they'll reach a point where they both, quote, climax, or whatever the equivalent is for frogs. And at that moment, the male releases sperm into the water, and the female releases a bunch of eggs into the water. So in the water, some of the sperm unite with the eggs, forming frog embryos. These embryos start to grow. And they grow, and it grows into something that looks like a fish. We commonly call it a tadpole or pollywog. And it looks like a fish. It doesn't have any legs, right? It has a tail, and so on. And then what happens is after it grows a certain amount, it starts to undergo metamorphosis. And it starts to not only develop legs that look like an adult frog, but the tail doesn't fall off. The tail atrophies. It shrinks. And so after about 3 or 4 months, what we now have is something that looks like a frog rather than a fish, like a tadpole. Now, of course, biologists have wondered for a long, long time what causes metamorphosis. Now, there were many, many hypotheses as to what caused a tadpole to begin changing into a frog. I'm not going to tell you all the wrong hypotheses. I'm going to tell you the one that turned out to be correct. So at some point, researchers suggested, they hypothesized, maybe the thyroid gland in the tadpole starts to release a bunch of thyroxine. And that increased release of thyroxine perhaps speeds up the rate of biochemical reactions in that tadpole, and that initiates all these biochemical changes as the tadpole metamorphosizes into a frog. Now, one of the ways they tested this hypothesis is they took a tadpole before it had grown larger. Normally, it would have grown larger, but before it had a chance to grow larger and undergo metamorphosis on its own, they injected it with thyroxine. And that immediately caused it to metamorphosize, so we ended up with a miniature frog. So that was one of the lines of proof of experimental evidence that giving the thyroxine was what triggered or initiated metamorphosis. Now, everything I've just told you was written right up here. So right above the picture, we wrote that the thyroxine hormone actually causes autolysis, right? Self-destruction in the cells of the tadpole's tail. So one of the things that thyroxine does is it doesn't cause the tail to fall off, it causes the cells to self-destruct. The lysosomes rupture, that's called autolysis, the cells self-destruct, and this tail atrophies or shrinks in size. So that's a third example of the role of lysosomes. Okay, let's return back to E3. So back on E3, I want to give you one last example. I don't think it's written there, but I've written it in. So I wrote letter D, visualize autolysis in cells infected with a virus. So in fact, when a virus enters human cells, infecting them, our body has to destroy these viral particles. We don't even have effective antiviral drugs. So one of the ways that our body causes these cells in our body that have become infected with the virus is that the lysosomes rupture, releasing those hydrolytic enzymes, destroying the cell and any viral particles within it. So in fact, what our body does is it basically causes some of our own cells that are infected with the virus to self-destruct. And that takes out the cell, but it also takes out the viruses that are inside the cell at the same time. And then we make new cells to take the place of those that have been self-destructed. The point here is this. The reason why there's so much emphasis in modern biology on cells is because this is how we explain everything. So you can see these silly little structures called lysosomes, and we're thinking, come on, this is like boring, who cares? I've just used lysosomes to explain how fingers form in a human embryo, how the womb of a woman atrophies or shrinks at menopause, how the tail atrophies during metamorphosis of a tadpole into a frog, and how the cells that are infected with a virus self-destruct to destroy the virus in our body. You have a question? Okay. So we're going to move on. Now, another vacuole, that's what we're dealing with right now, is vacuoles and vesicles. Another vacuole, a large sac, is found in all plant cells. And so those of you taking lab have learned this. There's something called a large plant cell central vacuole that stores water, nutrients, and waste products. Let's look at page E7, where we have a picture of this on the bottom left. So on the bottom left on E7, so it's got a picture of a plant cell. And looking at the plant cell, you can see labeled on the left-hand side, this is on the lower left of page E7, we see this large vacuole. It's huge. It's the largest structure in the whole plant cell. It's called a central vacuole that stores water, nutrients, and waste products. Very characteristic of plant cells, having that large central vacuole. All right, now back on page E3, what is a contractile vacuole? Now, contractile vacuoles, those of you taking lab are going to be learning about these in lab soon. Contractile vacuoles are structures, vacuoles or sacs, for the excretion of excess water that are found in all freshwater protozoans. So they're found in freshwater protozoans. Now, again, to help us visualize this, let's look at again at page E7. So on page E7, again, this time we're going to look at the top right. And on the top right, it shows a picture of an amoeba. An amoeba is a single-celled organism that lives in pond water. It is in kingdom Protista, which is made up of single-celled organisms that have a nucleus. And it is in phylum Protozoa. It is a protozoan, a single-celled organism that has a nucleus. It lives in pond water. It's kind of like the original blob, just kind of, crawls around in the pond water looking for food. And now, it's got a real problem, though. You'd say, tell me about it. What's its problem? I got problems, too. I want to remind you, pond water is essentially close to 100% water. But the inside of a cell is closer to 80% water. We've covered that previously. So if it's 80% water on the inside of the cell, but 100% water on the outside, is there a difference in concentration of water between the outside and inside of the cell? Yes. Yes. So which direction is water going to flow? Into the cell, right? Flowing from 100% water to the inside of the cell where it's less than 100% water. Now, this is called osmosis. And as the water flows in, if something doesn't compensate for this, the amoeba would just swell up and burst, but it doesn't burst. Why doesn't it burst? There is a special structure labeled right here called a contractile vacuole. And a contractile vacuole is a vacuolar sac that actually stores water. So as the water flows into the amoeba cell, that water accumulates in this sac called the contractile vacuole. When that sac fills up with enough water, it does exactly what its name says. It contracts. And when it contracts, it squirts the water back out into the pond. So we find that all freshwater protozoans, all protozoans, single-celled organisms that live in freshwater ponds, that all have this same problem because they are surrounded by 100% pure water, and they're less than 100% pure water, always have these adaptations called a contractile vacuole. We can see another example of this on your left. On your left, in the second row, is another protozoan called a paramecium. A paramecium, also in kingdom Protista, phylum Protozoa, just like the amoeba. Again, it's got the exact same problem. It lives in pond water, which is essentially 100% water. The inside of a cell is, well, it's mostly water. It's not 100% water. It's about 80% water. So as the water starts to flow into the cell, flowing from 100 to 80% water down its concentration gradient, and this is called osmosis, if something doesn't compensate for this, the cell is going to swell up and burst, but it doesn't. You'll notice labeled right here on the lower left, there is also a contractile vacuole here. In fact, there's another one right up here, contractile vacuole. And when they start out empty, they fill up with water, and when they become full, they contract, squirting the excess water out. So that is a unique adaptive structure found in all freshwater protozoans. Okay, that takes us to page E4. So on page E4, mitochondria. Now, mitochondria are found in cells. They're found in plant cells and animal cells, both. And they are nicknamed the powerhouses of the cell. Now, what's going on inside the mitochondria is a process called cellular respiration. This is going to be a big topic that we're going to cover for our third exam. But cellular respiration is the name of this process by which that gasoline called, and I haven't mentioned it yet, here it is, ATP is produced. ATP is going to be referenced every single class meeting. So make sure you know what it is for our next exam, because it's going to come up every single class. All right? So the production of this gasoline, or high-energy nucleotide called ATP, it occurs largely in the mitochondria of the cell. Let's look at a few pictures. And I made a note. We're going to look at pages E7, E10, and E12 to see what's going on. First off, on E7, this is the plant cell. And I just want to point out that mitochondria are found in plant cells. Here it is. It points to there are mitochondria in the plant cells. It's labeled on this is the plant cell on the bottom left, and it's labeled on the right-hand side. Now let's look at page E10. On page E10. All of the pictures on E10 are animal cells. Let's just look at the bottom picture. So on the bottom picture, this is an animal cell. It could also be a human cell. And you can see right here, labeled on the bottom right of E10, is a mitochondria. So they're found in plant cells, they're found in animal cells. Let's look at an enlarged view, so we can see this even better. Let's look at page E12. E12. So on page E12, if I can find my E12, I have too many pages in front of me. Yeah. Okay, on E12, at the very top left. So on the very top left, this shows an enlarged view of a mitochondrion, kind of a three-dimensional view. And a mitochondrion is characterized by having an outer membrane and an inner membrane. Incidentally, outer is just one T. But anyhow. So it has an outer membrane and an inner membrane. Right? So there's the outer membrane. The inner membrane has folded. You can see it has inner folds of the inner membrane. So it's folded. Now, it shows in this picture a whole bunch of little speckles or dots all over the inner membrane. Those speckles or spots represent enzymes. So there's a whole bunch of enzymes on the inner membrane of the mitochondria. Now, we know that enzymes are proteins that catalyze biochemical reactions. So there must be a lot of biochemical reactions occurring, especially on the inner membrane of the mitochondria. Now, in fact, the process that is occurring here, and I know that this looks like a mess, so don't worry about it. I'm going to walk you through all of this. Let's try to make it a little bit cleaner here as far as focusing. So what is occurring in the mitochondria is called cellular respiration. Now, the overall process of cellular respiration is what I've written. We're going to learn a lot more details about this in chapters 5 and 6. In cellular respiration, sugars such as glucose, C6H12O6, are broken apart using 6 molecules of oxygen, or O2. Now, there's really a whole lot of enzymes involved in breaking apart the sugar with oxygen. So it involves many, many enzymes for this process called cellular respiration. The sugars are broken apart with oxygen into 6 molecules of carbon dioxide and 6 molecules of water. And in the process of breaking apart the sugar, energy is released. And that energy that's released is used to make 38 molecules of ATP. Now, some books will say 36, but the point is we make all this gasoline called ATP. Is this the first time I've ever mentioned cellular respiration and the production of ATP? Not at all. We actually covered this back in section D. And if I just want to refer you back to page D14. Here's what I had written on D14. That this is D14. The way we make ATP is that phosphates are attached to ADP to make ATP, which is called a phosphorylation reaction. The energy that is used to phosphorylate ADP into ATP comes from the breakdown of foods, such as sugars, through a process called cellular respiration. So we're going to be learning a lot more about this. We're just learning the various parts of the puzzle that we still have to put together. All right, so we've been talking about the mitochondria. Let's go back to E4. And back on page E4, back on page E4, we had written that cells vary in their number of mitochondria. For example, in skin cells of our body, they have very few mitochondria. On the other hand, in muscle cells of our body, there are hundreds, maybe even thousands. How many mitochondria are found in a cell is an indication of how much energy it requires, because that's where this gasoline called ATP is made. So clearly, there's a lot of mitochondria in muscle cells because muscle cells require a lot more gasoline in the form of ATP to allow us to exercise and run compared to what skin cells need. So very curiously, we wrote that mitochondria contain their own DNA. Now, we've learned that there's DNA in the nucleus of a cell, but now we're telling you there's also what is called mitochondrial DNA in the mitochondria of the cell. I'll have more to say about that later. Now, what's listed next are plastids. Plastids. Plastids are sacs or vacuoles that are found only in plant cells. They're found only in plant cells. And there are 3 types of plastids: chloroplasts, chromoplasts, and leucoplasts. Let's start with the first one, chloroplasts, which are the most important of the 3. Chloroplasts, I've written, is where the process called photosynthesis occurs in plant cells. Now, photo means light, and synthesis means to manufacture. And what the plant cells are doing is, in the chloroplasts, is manufacturing sugars and other foods using light energy. So the synthesis of food, the production of foods like sugars using light, photo, is called photosynthesis, and that largely occurs in the chloroplasts. We're going to look at some pictures. Let's start with E4, and on page E4, if I can find mine. I'm sorry, sorry. Let's, yeah, we're going to take a look at E7. So let's look on E7. On page E7, on the bottom left, here's our plant cell. I'm being purposely redundant. And so here's our plant cell, and I just want to draw your attention to the fact that there are these green chloroplasts. In fact, the root chloro means green. They are colored green. Those of you in lab have seen chloroplasts in plant cells. All right, so these little green guys, right, are called chloroplasts. They're obviously in the plant cell. Let's see an enlarged view of the chloroplast by looking again at page E12. So on page E12, at the top right, so on page E12, we had a mitochondria on the left, kind of a three-dimensional view, and on the right, we have a three-dimensional view of a chloroplast. All right? Now, this is the top right on E12. Now, the reason why we've put the chloroplast right opposite the mitochondria is because there's some interesting similarities. Just like the mitochondria had an outer membrane and an inner membrane, so does the chloroplast. You'll notice it's labeled right here. There's an outer membrane, and right underneath it, there is an inner membrane. Right? They're labeled outer membrane, and right below it, I colored it red, is an inner membrane. It's not really red. Now, inside the chloroplast are these little flat discs. These flat discs, or sacs, are called thylakoid discs, or we'll just call them discs. And they form little stacks of discs. They kind of look like a stack of, well, we use the term pancakes for the Golgi complex, but they kind of look like pancakes also. Now, there's a lot of speckles or dots shown on those discs. Those speckles indicate, just like the speckles on the mitochondrial inner membrane, they represent enzymes. So there's a whole lot of enzymes in these little discs. Not only are there enzymes, there is the green-colored chemical called chlorophyll. So the green-colored chemical called chlorophyll, and chloro means green, is inside these discs, along with a bunch of enzymes. Now, the function of chlorophyll, as we're going to see in just a moment, is to absorb light energy. So the purpose of chlorophyll is to absorb light energy, and there's a whole bunch of enzymes for biochemical reactions. In addition, there is fluid surrounding these discs. Now, I tried to color it blue. I know it's hard to make out. But this fluid surrounding the discs is called stroma, and it's labeled right here. So what is stroma? I wrote that it is fluid surrounding the discs. And it also contains a lot of enzymes. So there's a lot of enzymes in this fluid surrounding the discs that's called stroma. So far we've said that there are these discs containing the green-colored chemical called chlorophyll, and enzymes, and there's a bunch of enzymes in the fluids surrounding the discs, and that fluid is called stroma. Now, the overall process that is occurring in the chloroplast is called photosynthesis. Let's see what photosynthesis is. Just to help make this cleaner, here is photosynthesis. Now, what is photosynthesis? Photosynthesis uses carbon dioxide, specifically 6 molecules of carbon dioxide and 6 molecules of water. Carbon dioxide and water molecules are joined together using sunlight energy, or light energy. It doesn't have to be from the sun, it could be from a light bulb. Now, what captures that light energy is the green chlorophyll. That absorbs the light energy. And there's a whole bunch of enzymes needed for this reaction. Just like in cellular respiration, there's a lot of enzymes because there's a lot of individual steps. And what happens is that this light energy that's absorbed by the chlorophyll is used to join carbon dioxide and water molecules together to form foods like sugars, foods like glucose. C6H12O6. Interestingly, a byproduct is formed. A byproduct is formed in photosynthesis of oxygen. So oxygen is actually given off or released by plants as they carry on photosynthesis. Now, some of you might be thinking, what, do we have to know this, do we have to memorize that? Well, you have to at least be able to recognize it. You don't have to write it on a blank sheet of paper, but we are going to ask you multiple choice questions about it. You'd say, yeah, but you're making us learn this. Do we also have to know that cellular respiration one? Yes, you also need to know this one. You'd say, so we got to know this one and this one? Yeah. Do you notice anything about the two? Aren't they the reverse of each other? You'll notice that in photosynthesis, carbon dioxide and water and energy are turned into sugar and oxygen. In cellular respiration, sugar and oxygen are turned into carbon dioxide, water, and energy in the form of this gasoline called ATP. Now, there's a lot more going on here. And what's going on is a very fantastic thing at the biochemical and cellular level, and also an amazing thing at the global level of the entire Earth, which we'll get into, because there's all reasons for all of this stuff that go deeper than where we're at yet. All right, so we've been talking about photosynthesis occurring in chloroplasts. Let's return back to the page where we're at, if I can find it, page E4. So on E4, under chloroplasts on page E4, so the chloroplast contains stacks of discs containing chlorophyll and enzyme. There is fluid surrounding the discs, also containing enzymes. That fluid is called stroma. The name of the process occurring inside the chloroplast is called photosynthesis, and its purpose is to produce foods like sugars. And very strangely, just like mitochondria, chloroplasts also contain their own DNA. So there's DNA not only in the nucleus of a plant cell, but also chloroplast DNA. We'll have more to say about that. Now, there's two other types of plastids. Plastids are these sacs only found in plant cells, not animal cells. What are chromoplasts? Now, chromoplasts are sacs that contain pigments other than chlorophyll. And I'm going to give you a couple of examples. So I'm just going to draw a picture here of a couple of things. Let me draw a flower. And since I'm drawing it fast, it's not going to be very good. And in fact, probably wouldn't even be good if I was drawing it slow. Let's say that it's a flower with red petals. Maybe it's a rose, even though it doesn't look like a rose. All right, now, our first question is, and I'm going to call it a rose, even though it doesn't look like it. All right, this is a red rose. All right, now, first question is, what the petals of the rose, these are petals. Do you think that the petals are made up of cells? Yes. They're not made up of aluminum foil. They're not made out of plastic or rubber. Every part of a living thing is made up of cells. Those of you taking lab have looked at an onion and seen that it was made up of cells. A leaf is made up of cells. Every part of the human body is cells. Brain cells, liver cells, skin cells, blood cells, lung cells, kidney cells, muscle cells. It's hard for us to imagine, but every part of a living thing is made up of cells. That's the cell theory. So this is actually made up of cells. If we enlarge this, if we looked at this with a magnifying lens, we would actually see cells. Plant cells typically are rectangular in shape. All right? And they would have a nucleus, and they would have a central vacuole that we've talked about. Now, here's the question, though. Do you think these cells that make up this red petals of the rose, do you think that they contain chloroplasts? No. No, and you're right. They do not contain green chloroplasts. Because if they did, the petals would look green. Now, when you look at a leaf is made up of plant cells with green chloroplasts, because the purpose of a leaf of a plant is to carry on photosynthesis. But the purpose of this, the flower of the red petals is for reproduction, not for photosynthesis. But what we would find is there are sacs inside these cells that would contain a red pigment. These are called chromoplasts. Now, chromo actually means color. So there would be sacs containing red pigments. These are chromoplasts. If in a leaf, we would find the cells would have green chloroplasts. So what gives it, the petals a red color are the fact that there is a red pigment in these chromoplasts of the cells that make up the petal. Let's give you a second very similar example. You'd say, what am I drawing? It's hard to know. It's supposed to be a carrot. Now, do you think carrots are orange? Yes. All right? I don't have orange, I got red, but okay. So, do you think that these cells, first of all, do you think a carrot is made up of cells? Yes, every part of a plant is made up of cells. So, do you think that these cells that make up the carrot contain those chloroplasts? No. We know it doesn't, because if it did, it'd be green. The question is, why not? Why doesn't the carrot, that part of the plant, contain green chloroplasts? Because this is part of the root, and it's under the ground. The carrot is, if you've ever grown a carrot, you've got to pull it up out of the dirt. So if the purpose of chloroplasts is a process called photosynthesis, you can't have a lot of photosynthesis without sunlight under the ground. This is a root, so it has nothing to do with carrying on photosynthesis. It's the leaves that look green, because the leaves are made up of cells with chloroplasts for the purpose of photosynthesis and making sugars. So this is made up of cells, but if we looked at these cells, they would look similar to these cells. There would be chromoplasts. They don't contain a red-colored chemical. They actually contain an orange-colored chemical called keratin. And so just explaining what chromoplasts are. So back on our page E4, so a chromoplast is simply a sac that contains pigments or colors other than green chlorophyll. It may contain the orange-colored chemical keratin, which would make it look orange. It might contain red-colored pigments or blue pigments, because there are flowers that are blue, flowers that are yellow, but it doesn't have green chlorophyll. Okay, now let's talk about a leucoplast. Now, leucoplasts are also known as amyloplasts, and the purpose of them is to store starch. Now, the reason why they're also known as amyloplasts is because the scientific name for starch is amylose. Did we ever learn that? Yes, page D2. D2. But they're also known as leucoplasts, coming from a Greek root leuco, meaning white, because starch kind of looks white. Let me try to give you an example of this. So, let me draw a picture. Again, it'll be amazing, I know. You'd say, what is that? This is a potato. All right? Now, a potato is part of a plant. Anybody know what part of the plant a potato is? The root. You pull up a potato up out of the soil, out of the ground. It's part of the root structure. So we understand why potatoes aren't green. Right? There are, white potatoes and brown potatoes. There's even some blue potatoes, right? But there's no green potatoes, because you can't do photosynthesis under the ground. Now, it is made up of cells. If we looked at these with a magnifying lens, we would see the potato, just like a carrot, is made up of cells. There is a nucleus. There is a large central vacuole. but there are also sacs that store starch. All right, so I'm just coloring it yellow, but these are called amyloplasts or leucoplasts. An amyloplast means that it stores amylose or starch. So these are known as amyloplasts. They store starch. So would that make sense then that if the cells that make up a potato contain amylose or starch, would that explain then why a potato is high in starch or amylose? The purpose of a potato is it stores these sugars, right, as a polysaccharide called starch or amylose in its root. Incidentally, just thinking back for a moment about that carrot, carrots are kind of sweet. So what do you think is stored in the cells of that carrot? Sugars. That's why it tastes sweet. So commonly, many plants store sugars either as sugar or as starch, which is just a bunch of glucoses joined together, and they store these foods in their roots. That's what an onion is. Incidentally, the onion, which has a characteristic taste of an onion, but interestingly, if you fry up the onions, don't they start to taste sweet? Because actually, there's a lot of sugar in onions. All right? If you cook them, then you don't become less aware of that biting taste of the onion, and you start to actually taste sweet. They become caramelized, as it were. Right? So that's also part of the root, the onion bulb. So this is very common. How about beets? Anybody know, those red beets? That's where we get sugar also. They're called sugar beets. That's the root. Sugar is stored in this. So a lot of plants store sugars or starch in the roots. All right, we've been talking about these plastids in plant cells. Now we're going to, the last few structures we're going to tackle kind of simultaneously. And these are centrioles, flagella, and cilia. Okay, this is page E4. Now, the centrioles we wrote are found only in animal cells, and they are involved in cell division. They are made up of microtubules, and specifically a ring of 9 sets of microtubules that are capable of contracting. All right? Then, a flagella or cilia. So are you going to clarify that? Yes, I am. The flagellum is a whip-like tail that is made up of 9 paired sets of protein cylinders or microtubules, plus 1 pair in the center. You say, I don't get that. We're going to see a picture of this in a moment. It looks like this. This is 9 pairs of microtubular proteins, tube-shaped proteins with a pair in the center. This is known as a 9+2 arrangement of microtubules. All right, and then a cilia. Cilia are fine motile hairs that are organized just like flagellum. All right, well here, let's try to clarify some of this. First, let's start with a flagellum. A flagellum is a tail. I wrote that right here. So, an example of a flagellated cell, a cell that has a flagellum, are human sperm. The flagellum acts like a tail and it moves. And as it moves, it propels the sperm through a fluid environment. All right? Now, this shows a paramecium, which we've seen pictures of before. And paramecium is a single-celled organism, a protozoan, that has little cilia. Now, really, what cilia are is like short flagellum. It's like a little tiny short flagellum. And they move. So we can see that flagellum, which create movement, and cilia, which create movement, are all associated with movement. Let's take a look at some pictures of this on page here. I think it's E12. Yeah, E12. So on page E12, on the bottom left, on E12, so it shows a flagellated cell. Could be a sperm. Could be another cell that's flagellated. Sperm are not the only flagellated cells. They're only human flagellated cells, but There are all kinds of single-celled organisms that have flagella. And if we made a cross-section to see what's inside the flagellum, it would look like this. It would have 9 pairs of microtubules around the perimeter plus 2 in the center. It would look like this model. This is called a 9+2 array or arrangement of microtubules. Now, these are cilia. Cilia are like short flagellum. If we made a cross-section through them, it would look absolutely identical to the inside of a flagellum, a 9+2 arrangement. So what allows a flagellum to move, or cilia to move, is that these microtubule-shaped proteins move. That is the basic unit of movement. We actually learned about microtubules previously on page, I believe it was E1, E2, I'm sorry, page E2, where we talked about cytoplasm, and we said in the cytoplasm there are microtubular-shaped proteins and microfilamentous proteins, and the tubule-shaped ones create movement. Now, just above is a centriole. I didn't bring my model of a centriole, but a centriole looks very similar to a flagellum or a cilium. It has 9 sets of microtubules around the perimeter, but it's missing the pair in the center. This is called a 9+0 arrangement. Now, the centrioles are structures that are involved in creating movement in animal cells when they divide. So in a lab class, you learn that when you learn about how cells divide, you learn that in animal cells, including human cells, there are these centrioles that form these microtubules that extend between the centrioles. This is part of what's called the mitotic apparatus inside of an animal cell. And these microtubules actually pull the chromatids or DNA molecules apart during cell division. So let's go back to E4. Back on page E4, what's the point we're trying to make? Centrioles, which are found only in animal cells, create movement. They are involved in movement during cell division, at least in animal cells. It's these microtubules that create movement. The flagellum. There are cells, including human sperm, that have a tail or flagellum that moves, and what's inside them are microtubules. There are cells that have cilia, these cilia are like little hairs that move, and what's inside them are microtubules. So clearly, the basic unit of movement are microtubules inside the flagellum, inside the cilia, or inside the centriole. Now, just before we leave this, we've already mentioned an example of a human cell that it has a flagellum are sperm. And incidentally, the purpose of the cilia in our airways is not to make the cells move, but to push dust and foreign matter out of our airways to keep them clean. So the cells, the ciliated cells that line our trachea, our windpipe, that line our bronchi and bronchioles, they have cilia, and these cilia move back and forth to push the microscopic dust and foreign matter that we've inhaled. Every time we inhale, it pushes it back up out of the airways, up into our throat. You can imagine every time we inhale the amount of little tiny microscopic bits of dust that we end up inhaling. So we've got to keep our airways clean. Now you might say, well, if the cilia push it up into our throat, what do we do then? We do one of two things. We either go and spit it out, or we swallow it, and whatever we swallow, if it's just dust and stuff, it'll come out in our feces or stool. But the idea is to keep our lungs clean. Now a second place where we have ciliated cells, and I don't think any of you would have guessed this unless you've learned it, is in the fallopian tubes of a woman. Now the fallopian tubes are also known as the oviducts. And so to help us understand this, let's take a quick look at page L8. L8. You might say, what's on L8? Section L, page 8. At the top of L8, we're looking at the female reproductive system. This is the female reproductive system. And so what we have here is these are the ovaries. Here's an ovary, here's an ovary. This ovary on your right is actually a cutaway view. And we can even see an egg popping out. When an egg pops out, that's called ovulation. Now, these are the fallopian tubes or oviducts. And normally when this egg pops out, it actually enters the fallopian tube. Now your question might be, well, if the egg comes out here, how the hell does it get over to the fallopian tube? In real life, these fallopian tubes are attached to the ovaries. So in this picture, they drew the fallopian tubes pulled away from the ovaries, but in real life, they are actually attached to the ovary. So when that egg pops out of the ovary, it enters the fallopian tube. Interestingly, though, an egg cannot move on its own. A sperm can, but not an egg. This egg that enters the fallopian tube and pops out is going to be pushed by ciliated cells lining the inside of the fallopian tube. So there are the cells that line the inside of the fallopian tube, slowly but surely push that egg through the fallopian tube towards the uterus or womb. The uterus is the womb. Now it takes about a week for this egg to be pushed through the fallopian tube to the uterus. That is the 1 week out of every month where a woman can get pregnant. A woman cannot get pregnant when the egg is still inside her ovary. But that egg only lives about 1 week and then it dies. Sperm will live about 3 days inside of a woman's body and then they die. But the egg will only live about a week, it's really less than a week. So if a woman's going to get pregnant, the sperm, the only place where she can get pregnant is where the sperm unites with the egg as the egg is being slowly pushed through the fallopian tube in the direction of the uterus. So in fact, let's look at L9. On L9, page L9, so here it shows exactly what we said on L9. The egg is being fertilized by a sperm in the fallopian tube. That's where fertilization or conception usually occurs. That forms a zygote, a fertilized egg, and it starts to develop. A 2-celled embryo, a 4-celled embryo, an 8-celled embryo. And again, the embryo cannot move on its own either. It's being pushed by ciliated cells that line the inside of the fallopian tube. Again, whether it's the egg or whether it's the embryo, it is being pushed through the fallopian tube, and if it's an embryo, once it reaches the uterus, it will implant in the inner lining of the uterus. The inner lining of the uterus is called the endometrium. Endo means inner, metrium means uterus. The embryo will implant in the inner lining of the uterus, the endometrium, and it will be nourished by blood vessels in the endometrium of the uterus. Now, one last thing here. Has anybody ever heard of something called a tubal pregnancy? So, just to write down the term, tubal pregnancy, also known as an ectopic pregnancy. Now, what's that? Imagine if the egg gets fertilized, so we have an embryo, but what if there's no ciliated cells? What if the cells lining the fallopian tube either do not have cilia, or the cilia simply don't function? So then, there's no cilia to push the embryo to the uterus, and it just starts to develop right there in the fallopian tube. Now, they have to terminate this pregnancy, abort it, because obviously the fallopian tube is not designed to accommodate a baby to grow in it, and it was just going to rupture the fallopian tube, causing hemorrhaging or bleeding, and the embryo is not going to be able to survive anyhow. So one of the possible causes of a tubal pregnancy is not having those ciliated cells, or cells with functional cilia. So, in summarizing today, we were talking about microtubules just now. Microtubules are the basic unit of movement that are